It began with a discovery that forces us to rethink who we are and where we came from. From bones more than 45,000 years old, scientists coaxed out the oldest human DNA ever read, a whisper carried across deep time. Like this kind of story? Subscribe so you won't miss the next chapter. For decades, the early pages of human history seemed sealed shut. Bones and tools offered clues, but our ancestors' voices were silent. DNA, the fragile code of life, crumbles beyond a certain horizon. New techniques have cracked that barrier. Complete genomes from Europe's earliest modern humans, vivid enough to show whom they met, how they mixed with Neanderthals, and why their lineage ultimately disappeared. Before genetics, prehistory spoke in fragments. A napped flake here, a hearth stain there, a weathered skull whose contours hinted at kinship. Typologies and radiocarbon dates sketched timelines, but they couldn't resolve intimate questions. Who these people were, whether two sites held members of the same community, or how far travelers carried ideas and genes. Culture could spread without people, people could move without culture, and bones alone seldom settled the debate. The result was a map of possibilities, elegant, suggestive, and fundamentally mute about ancestry. DNA promised answers but imposed a harsh limit. Heat, moisture, microbes, and time chopped genomes into dust. Beyond a threshold, the molecular letters are too few and too damaged to read. Early ancient DNA work delivered only tiny glimpses, short markers that hinted at kinship yet left the story incomplete. Contamination lurked everywhere. A modern fingerprint could drown out an Ice Age signal. For years, the consensus was pragmatic skepticism. Past a certain age, genetics would never speak. The border of deep time seemed fixed until it wasn't. In 1950, excavators lifted a fractured but remarkably complete skull of a young woman from a limestone hill called Zlati Kun, catalogued, measured, and then largely consigned to quiet shelves. For decades, it spoke only through shape: brow curve, vault thickness, the geometry of a face. No one imagined that its dense bone still cradled molecules resilient enough to outlast Ice Age winters and humid summers. When researchers later revisited the specimen with ultra-clean protocols, they found what earlier science could not: a reservoir of ancient DNA, long silent, waiting to be read. A few decades later, an unexpected companion joined the story. A 45,000-year-old human bone from Siberia, followed by jaws and teeth from a cave site known as Bacho Kiro. Teeth proved ideal, compact time capsules, their inner dentine sealed from the elements. Drilled to fine powder and processed under severe contamination controls, they yielded genetic fragments once deemed unrecoverable. Piece by piece, those shards aligned into legible sequences. What had been a scattering of clues, bone here, tool there, suddenly acquired a voice, as if the Ice Age had cleared its throat and started to speak. Ancient genomes don't arrive as tidy strands. They're chemical confetti, tens of millions of ultra-short fragments, many touched by time's edits. Laboratories rethought everything: sterile, clean rooms, bleach, UV, and positive pressure air. Double gloves and suits to keep modern DNA out. Teeth and dense petrous bone were drilled to fine powder, then incubated so broken molecules would slip free. Enzymes trimmed damaged ends. Bar-coded adapters latched on, letting machines capture even 35 to 40 base pair slivers. Computational filters then separated true Ice Age molecules from microbial noise and modern contamination, mapping each shard against a reference to rebuild the sequence base by base. For years, results were glimpses: mitochondrial lineages, a handful of nuclear markers, patterns suggestive but incomplete. Then capture techniques and deeper sequencing changed the scale. Instead of sampling a few informative sites, teams retrieved coverage across the entire genome, enough to phase segments, estimated mixture, and time events via recombination clocks. Teeth from Zlati Kun, Bacho Kiro, and other early sites no longer yielded whispers but full scores. Readable genetic blueprints of people living more than 45,000 years ago. With coverage came precision. Not just they were related, but how closely. Not just there was mixing, but when it happened and how the resulting ancestry threads wove or broke through Europe's earliest human story. Compared with us, these early Europeans carried far larger chunks of Neanderthal DNA, long, unbroken tracts running through their chromosomes. Recombination, the genetic shuffler that slices segments a little shorter each generation, hadn't yet had time to chop those pieces small. That length matters. It's a ruler for deep time. Where present-day people show a fine dusting of short Neanderthal fragments, these genomes display bold strokes, a sign that the encounter between lineages sat only a short distance behind them on the family tree. Measured against recombination's clock, the data place these individuals just a few hundred generations after the first mixing of modern humans and Neanderthals. In other words, we are looking almost directly at the wake of that meeting, close enough that its genetic ripples still roll intact across their DNA. With whole genomes, scientists could quantify not only that admixture happened, but roughly when and how its legacy began to dilute as later populations arrived. The result is not a mythic episode, but a datable event etched into real people's cells. The shock was not that these people were early, it was that they are not us. When their genomes are compared to present-day Europeans, the fit is poor. Shared ancestry is minimal, and key lineages trail off rather than thread forward. The simplest reading is stark. An early wave entered Europe and left little lasting mark. Climate swings, competition, small founder groups, and sheer demographic chance could all have pushed their line to the margins. They opened paths, survived for a time, and then their branch withered. This finding reframes Europe's peopling as a sequence of waves, not a single founding surge. Some streams joined and swelled, others split into side channels that dried up. Later arrivals, genetically distinct yet related, overwrote much of the earlier signal, while a thin residue lingered where histories overlapped. The metaphor matters. Ancestry is not a straight line, but a braid, with currents merging, diverging, and sometimes disappearing. To understand who we are, we have to see the absences too, the lost threads that shaped the channels we still carry. These were small bands stretched thin across steppe and forest, moving with game and seasons, caching stone and fire in caves that doubled as archives. Winters were long, protein-rich diets unforgiving, and injuries costly when skilled hands were few. Neanderthals still occupied parts of the continent. Predators stalked the same corridors. In such landscapes, chance loomed large. A bad year, a failed birth cohort, or a blocked migration corridor could erase a lineage outright. Genomes preserve these pressures as faint seams, signs of bottlenecks where diversity pinched, hints of kin-structured groups, and admixture tracts reflecting encounters, friendly, neutral, or desperate. Short runs of homozygosity flag episodes of isolation. Longer ones betray close kin matings in tiny populations. Layered on top are the Neanderthal segments, still long in these early people, shrinking in later groups as recombination and fresh arrivals diluted them. The result reads like a ledger of risk, resilience, and attrition. Paradoxically, their absence from our genomes sharpens the picture. Because these early Europeans didn't flow into later populations, their DNA preserves a clean snapshot of Europe's dawn. Admixtures still fresh, demographic scars unblurred by later waves. They serve as temporal anchors, fixed points that let researchers calibrate clocks, test models, and separate cause from coincidence. In their genomes, we catch a rare, still frame of a continent mid-transition before subsequent migrations smeared the lines. If one entire lineage can vanish, how many more are missing from our reconstructions? Every dried channel in the ancestral braid implies stories untold, languages never recorded, tool traditions that flickered out, names no one spoke again. Ancient genomics turns that loss into knowledge, not to mourn alone, but to map the forces that pruned and shaped us. Seeing the gaps changes the questions we ask about who remained and why. What moves us here isn't myth, but molecules. Real people, their genomes carried across 45 millennia, speaking in a language of segments and seams. They walked into a cold continent, met others like and unlike themselves, and then their branch ended. Yet in ending, it illuminated the path. Ancient DNA lets us hear them again, the surge of arrival, the hush of disappearance, the faint harmony where lineages crossed. Humanity is not a single melody, but a braided chorus, some voices fading, others rising. Each new tooth, each quiet bone, is a chance to tune the ear and rewrite the opening bars. Thank you for joining this journey into the deep past. These discoveries remind us how fragile and yet how enduring our story truly is. Every ancient genome adds another voice to the chorus of humanity, and together they bring us closer to understanding who we are. If you enjoyed this exploration, consider subscribing and sharing. It helps keep the story alive. Until next time.